Stability of foreign proteins in E. coli. So in addition to other factors that are important, so this is also one of the major aspects that we must take into account while cloning foreign DNA for the isolation of specific protein product, that is how much it is stable. So this problem of instability of foreign proteins, it is well recognized when we use bacteria as a host organism, because although it is expressed, but it is also degraded or become unstable at the same time. So this is also one of the major issue about the stability of the foreign proteins in a particular host organism. It also has been reported that this stability of the foreign protein or even the native proteins, it is also dependent upon the location within the cell. For example, ये देखा गया है कि अगर प्रोटीन्स जो हैं ये साइटोप्लाज्म में हों तो उनकी हाफ लाइफ जो है वो 2 मिनट तक होती है, और अगर वो पेरिप्लाज्म में हों तो उनकी हाफ लाइफ जो है वो 10 फोल्ड इंक्रीज कर जाती है. So the location of the expressed protein, it is also important. So different strategies has been used to tackle this problem, that is instability of the foreign proteins in the host organism. And one of the strategy, as I told you, that it is the expression of proteins in the form of fusion proteins. For example, here one can quote the example of proinsulin uh, in combination with beta galactosidase. So rat proinsulin, it can be expressed as fusion proteins with an enzyme, that is beta galactosidase. So in the form of fusion proteins, their stability it is increased, and later on the native protein, it can be cleaved with the help of a chemical like cyanogen bromide can be used to get the expressed protein from the fusion protein. So this is one of the strategy. But the overall problem that is related to the instability of the foreign protein, it is because of the protease system of the host organism. Because if host organism it contain proteases, then the proteins that are expressed as a foreign proteins, they may lack three-dimensional structure or the tertiary structure. They are not in a properly folded state at the beginning. So the host proteases can recognize them that they are abnormal proteins, and then they are degraded immediately. Now this problem, it can be solved if we use host organisms that are deficient from proteases. For example, in case of a lone protease, that is very famous class of proteases, LON, or lone protease. They are ATP dependent proteases, and they can target the unfolded uh, proteins within the cytoplasm, and they can degrade them into individual amino acids. And here in this case of lone protease, the activity it is ATP dependent. In addition to that, if we want to get the this foreign expressed protein, then the host organisms that are deficient to lone protease, they are more advantageous. For example, uh, lone protease deficient host organism has been constructed, but again there is problem because the lone deficient protease microbes, they are showing more mucoid growth that may be problematic during the culturing of the recombinant microbes. In addition to that, there is extensive filamentous growth of the host organism that may be non-viable. This problem has been overcome by using another mutant that is inactivating the gene which is called as the GOL-E, uh, the, that play role in the biosynthesis of the capsule. And in addition to that, the cultural conditions uh, can be modified so that the mucoid growth can be stopped. For example, the yeast extract component of the culture media, it can be removed. In addition to that, if we want more stable foreign proteins, then the microbes that are deficient of sigma 32 factor that mediate heat shock response, then it can be used to increase the accumulation of foreign protein. Another factor or protease that is important, it is CLP protease. It is also playing role in the degradation of different types of proteins. Either they are the abnormal proteins or even the regulator proteolysis, where the normal proteins under starvation, they can also be degraded. So if we create the mutants that are deficient in proteases, especially lone or CLP proteases, then the accumulation of the foreign proteins, it can be enhanced. Another factor, as I told you, that the cultural conditions of the host organism, it is also very important. क्योंकि अगर हम कल्चरल कंडीशन को कंट्रोल करेंगे तो प्रोटियोलाइसिस या फॉरेन प्रोटीन्स की उसको भी कंट्रोल किया जा सकता है. मिसाल के तौर पर अगर कल्चर मीडिया में न्यूट्रिएंट्स नहीं है, स्टारवेशन है, तो प्रोटीन्स की डिग्रेडेशन रैपिडली होती है. अगर हम नाइट्रोजन का सोर्स या कार्बन सोर्स को कंट्रोल करें तो जो रेगुलेटर प्रोटियोलाइसिस है उसको कंट्रोल किया जा सकता है. तो इस पर्पस के लिए अगर हम बायोरिएक्टर्स यूज़ करें that are commonly used for the cultivation of microbes in large quantities, then we can adjust such cultural conditions that are related to nutrition, pH, oxygen, or other gaseous requirements.